Lung cancer is the leading cause of cancer death worldwide. Each year, about 2.5 million people are diagnosed, and most will die from the disease. The most effective way to prevent lung cancer is not to smoke. Tobacco use causes the vast majority of cases, but there is another powerful opportunity to reduce deaths: early detection. Screening with low-dose CT scans can detect lung cancer at an earlier stage, when treatment is more likely to be curative. Large studies have shown that screening saves lives, but only when it is targeted to groups of people where lung cancer is more frequent. In other words, people at high risk. That raises a crucial question. How do we decide who should be screened? Today, screening eligibility is mainly based on age and smoking history. While these criteria are important, they are far from perfect. Many people who go on to develop lung cancer are missed, while others who qualify for screening have low risk and are unlikely to benefit. This is where the Integral project comes in. The idea was simple but powerful. Could a blood test help identify people who are most likely to develop lung cancer in the near future? To answer this, the International Agency for Research on Cancer brought together researchers from across the world to analyze blood samples from thousands of healthy volunteers. These samples had been donated years earlier, before anyone knew who would eventually develop lung cancer. The study unfolded in 3 steps. First, researchers scanned over 1,000 blood proteins to identify a small set linked to future lung cancer risk. Next, they put these proteins on a custom assay and built a prediction algorithm called Integral Risk. This algorithm combines age, smoking history, and the levels of 13 blood proteins to estimate a person's risk of developing lung cancer. Finally, in a completely new set of participants, the team tested how well this model could predict future disease. The results were striking. Integral Risk performed far better than existing approaches at distinguishing between people who would soon develop lung cancer and those who would not. Most importantly, this improvement had real consequences for screening. When comparing different methods, while referring a similar number of people for CT scans, the Integral Risk model identified 85% of lung cancers diagnosed within the following year. Current criteria identified only 63%. In simple terms, the blood-based model found many more people who were on the verge of developing lung cancer. This is not about replacing CT scans. It is about making smarter decisions before the scan even happens, targeting screening to those who need it most, missing fewer cancers, and ultimately saving more lives. These results are encouraging and could lead to more people being cured from lung cancer in the future. They highlight what is possible through rigorous science and international collaboration.